Good morning, everyone. It is an absolute pleasure to be here with you, although virtually. My name is Farid. I'm a new faculty at the New Jersey Institute of Technology, and today I'm going to present some of the work I did during my postdoc in Shenoy Lab at the University of Pennsylvania. The topic of our work is mechanobiology, and I think it is a new field for many of you. So let me start with a brief introduction to mechanobiology and its role in health and diseases. I guess we all know Robert Hooke because he was the first to visualize microorganisms and he was the one who coined the term cell. But Robert Hooke is also a very well-known scientist in the field of mechanics and physics because in 1660 he discovered the most fundamental law of mechanics, which is called Hookean law. But I think even Robert Hooke himself did not expect any connections between his two discoveries. But after more than 3 centuries, we as mechanobiologists are now studying the interdependence of Hooke's discoveries, the interdependence of biology and mechanics, or in other words, how mechanical factors, including stiffness, geometry, and force, can impact different cellular behavior like proliferation, differentiation, and gene expression. And I think the best example to describe mechanobiology is cell differentiation. In the body, obviously, we have different organs, and each organ has its own cells. For example, in the brain, we have neuron cells, or in fat tissues, we have adipocyte cells, and so on. And we know that all these different types of cells are formed by stem cells. For example, stem cells can differentiate and become neuron cells, or they can become osteocyte cells or any other cell type. But what is interesting is that mechanics can be an important factor in cell differentiation. So here, I'm going to show you how mechanics can influence cell differentiation. Here in this example, we have stem cells cultured on hydrogels with different stiffness, but under the same chemical environment. In the first case, cells are cultured on a very soft matrix that mimics the stiffness of the brain. In the second and third cases, cells are cultured on stiffer matrices with the stiffness of typical fat and muscle. And in the last case, cells are cultured on a rigid substrate which represent the mechanical environment of bone tissues. Interestingly, you can see that cells on the softest matrix become neurons, cells on stiffer matrices become adipocyte and myocyte cells, and cells on the rigid substrate become osteocytes. So this example clearly shows that the stiffness of the extracellular environment matters, and that stiffness can determine cell fate. And it is not just stiffness. Other mechanical factors like geometry and force have been also shown to impact cell behavior. For example, if we change the cell geometry and confine it to a small area, or in other words, if we do not let the cell spread freely, then we will see cell apoptosis. So this example tells us that geometry is important and can control cell life. Or if we apply mechanical forces to a cell, for example, if we stretch a cell, as you can see in this example, then we will see many changes in the cell, including changes in the cytoskeletal organization. So the take-home message is that mechanical signals like stiffness, geometry, and force, they all can change cell behavior. With that short introduction, I'm going to briefly present some of the work I did on the mechanobiology of cells and tissues. And I'm going to start with extracellular forces. We know that cells in the body experience external forces in the form of tension, compression, and shear. These external forces sometimes can damage cells, like in brain injuries and concussions, and sometimes they positively impact cellular behavior, like the positive effects of cyclic forces during physical exercises on bone and muscle tissues. A great example of cells under external forces is the human ocular surface. Each time we blink, cells on the ocular surface experience external forces. But what is important is the fact that these external forces are necessary for the normal function of cells, which means that any alterations in these physical forces, for example, because of diseases such as dry eye disease, can lead to abnormal cell behavior and even inflammatory responses. So there is a great interest in making artificial tear films that can restore the original force field in patients with dry eye disease. To this end, eye-on-a-chip systems can be a good platform to test the effect of different drugs. But one of the first steps in designing such systems is to ensure that cells in our systems experience the same level of physical stress as cells experience in the human eye. And this is where we need a theoretical model. So without going into details, we developed theoretical models for the ocular surface, for the tear film, and for the eyelid. And then by coupling these models, we could determine the complex physical stress field of the ocular surface, including the pressure and shear stress that cells experience during blinking. And that enabled us to design an eye-on-a-chip system that can faithfully recapitulate the biomechanical environment of the human ocular surface. After talking about external forces, I'm going to talk a little bit about internal forces. We know that cells in the body not only experience external forces, but also generate forces on their own. One of the earliest pieces of evidence was presented in this science paper, showing that cells cultured on a soft polymer would pull on the polymer and wrinkle the surface. This indicates that cells can generate forces and they can transmit these forces to their surrounding environment. And now, after almost 2 decades, we know that these internal forces are absolutely essential for many biological processes. For example, if we think of a cell as a car, these forces are the engine. Cells use these forces to migrate. Cells also use these forces to close the wound. For example, during the process of wound healing, normal fibroblast cells, as you can see, first become myofibroblast cells, which are more active and more contractile cells. Then these cells migrate toward the wound site and they deposit collagen. And then by generating high contractile forces, they close the wound. But after the closure of the wound, if this force generation continues and cells do not return to their original state, then it can lead to scar formation. And in more serious cases, it can even lead to formation of fibrotic tissues. So internal forces can have both positive and negative roles in the body. And of course, the worst negative effect of cellular forces is their involvement in cancer progression. Cancer cells use these forces to escape from the primary tumor and invade other organs. So it is really important to measure these forces. And here is the specific question that by measuring cellular forces we wanted to answer. We know that the physical properties of tissue change during aging or after injuries or diseases such as diabetes. We know that in all cases, the tissue becomes stiffer. So the question is how these changes impact the behavior of cancer cells. Do cancer cells become more invasive or less invasive? To answer this question, we need to measure cellular forces. But how we can do that? First, we need to culture cells in 3D collagen networks. And as you may see, we have small fluorescent beads that are attached to collagen fibers. So by tracking these beads, we can determine the displacement field in the collagen matrix generated by cells. Then to measure force, we need to develop a theoretical model that gets the displacement field as an input and then calculates force. So without going into details, we develop such a theoretical model and we use this model to measure cellular forces generated by MDA breast cancer cells. As you can see, we have an array of 9 collagen matrices. These collagen matrices have been made with different collagen concentrations, different temperatures of polymerization, and also different levels of cross-linking. So as you can see, they have totally different microstructures. And by measuring their stiffness, we also found that they have different stiffness. Some of them are very soft, some of them have the stiffness of normal tissues, and some of them are very stiff and show the stiffness of aged tissues or tissues after injury. And also some of them uh, have high levels of glycation, so they can represent tissues in a person with diabetes. So using the theoretical matrix model and without going into details, we measure cellular forces in each of these 9 collagen matrices. And then we plotted the force as a function of collagen stiffness. And interestingly, we found that cancer cells generate higher forces in stiffer collagen matrices. And why it is important? First of all, we know that collagen stiffness increases after injury or with aging or with diseases such as fibrosis and diabetes. So this means that MDA breast cancer cells become more aggressive in people with previous injuries or in older people or in people with diseases such as fibrosis and diabetes. And that may explain why chronic wounds, aging, and diseases such as fibrosis and diabetes can increase the risk of breast cancer invasion. As my last example, I'm going to talk about the interdependence of internal and external forces. For example, consider fibroblast cells in connective tissues. We already talked about internal forces, and we mentioned that fibroblasts use these internal forces to close the wound during the process of wound healing. We also mentioned that these internal forces can be the main cause of fibrosis. On the other hand, we know that fibroblasts in the body can experience external forces during daily physical activities. For example, when we sleep, these cells experience compression. So the question is how these compressive forces affect cell behavior and more specifically cell internal forces. To answer this question, we can culture a fibroblast on a rigid substrate and then put a plate on the top of the cell and apply compressive forces. But before showing the results, let me first talk a little bit about the cell cytoskeleton. The cell cytoskeleton is composed of several components. The first element is actin that can polymerize or depolymerize continuously. The second component is myosin that can attach to actin and generate force dipoles. And that is how cells can generate internal forces and how cells can contract. The next component is microtubules that usually experience compression as the cell contracts. And the last component is intermediate filaments. Now, let's get back to our problem and see what will happen to the cytoskeleton of a fibroblast cell when the cell experiences compressive forces. We can see that under compression, actin filaments are depolymerized and the level of myosin phosphorylation significantly decreases, which means that under compression, cells become low contractile and generate less internal forces. So the next question is, what is the consequence of this reduction in actomyosin contractility? Well, we can see that under compression, histone deacetylates or HDAC translocates from the cytoplasm to the nucleus. So cells under compression have a higher level of HDAC inside the nucleus. And we know that the role of HDAC is to condense chromatin. And this is exactly what we see. We see that cells under compression have higher chromatin condensation and they become transcriptionally less active. But interestingly, we see that when we remove the load, cells get back to their original conditions and they will have again their normal gene transcription pattern. So this tells us that as long as we are sleeping and compressing fibroblasts, these cells cannot have their normal gene transcription pattern. But when we release the load, they can get back to their normal condition. So an interesting question for future work is that what will happen if we keep these forces for a very long time? For example, in a person with a severe disability that cannot move or roll over for a very long time. So with that, I finish my presentation. I would like to thank my collaborators and also funding sources. Please also feel free to contact me if you have any questions or suggestions. You can see my email address and also my Twitter address here. So thank you very much for your attention and I hope you have a nice rest of the day.